Good morning. Next one. Oh. I think in 2004, it is important to remember the two things. That one, this Eve has already mentioned, the gaps, the total lack of drugs for the treatment of these diseases. The other thing that he hasn't mentioned is the frustration that was felt by many research scientists in the community who had good ideas, who had compounds. There was nowhere to go. And so the development and the growth of DNDi is not just about filling those gaps. It's about also being able to engage with the community, research community throughout the world, to relieve that frustration and ensure that things can move forward and people can identify with a pathway of developing new treatments. And so, back in those days, when we actually sat down and following on from the DND, the working group, and sat down to actually think about how to move forward, it was quite clear that there were opportunities there. The opportunities which came from the understanding that there were many researchers around the world who had novel ideas to exploit, but also some of the gaps, gap 2 and gap 3, where really there were already low-hanging fruit. There were potentials for clinical trials and potentials for new formulations, and we'll come on to those in a minute. But the big opportunity was to establish with the research community a long-term goal of developing cheap, oral, safe drugs for the treatment of these diseases. And when we actually came to the start in 2003, 2004, there was already some initial calls for the programs and for projects. Through the international press, calls were made for people to come forward, submit projects, and there was a review system through the Scientific Advisory Committee, which Diane Worth was the chair of at that time, and programs and projects were selected to move forward. But I think it really became clear very early on that though there were many good and excellent ideas coming from the academic community, we were missing a major opportunity to engage with the biotech and pharmaceutical sector, where there were already a lot of projects that could be used, that could be engaged with, and actually there was a lot of knowledge in the biotech and pharmaceutical sector that actually knew better than us in many ways how to develop drugs and get them through. And so it was actually within really the first year, from 2004 to 2005, that the R&D team, as it became known within DNDi, started to grow and develop, but also to change the pattern of work and actually move from a reactive mode, reacting to the calls, but into a proactive mode to actually engage with the biotech sector and with industry. And I say this came about through a teamwork, and this was something I'll come back to right at the end. It's very important to understand the nature of building a group of people within DNDi to do this work. So the decision was made to look at all three opportunities, the short term, the medium term, and the long term. But obviously to get a new drug out, as we know, and it's said in the literature many times, is a 10-year project at least. How do we start? How do we get there? Well, the next slide, so it illustrates at the bottom some of the early proactive projects that were actually started really back in 2005. But the slide, which it actually comes from a presentation I made in 2007 towards the end of the period, is here to illustrate really just two things. This was just for the slide for African trypanosomiasis, not for leishmaniasis or Chagas disease. And the point here was, how do we actually then engage with the community and with the sectors to develop new drugs, knowing the complexity of discovery, preclinical and clinical development. And the messages really were twofold. One, that knowing the attrition rate that is implicit within drug development, there's got to be things that don't succeed, and there's a high rate of projects that don't succeed. Not all chemistry is good. Not all biological systems that measure that chemistry are good. So we have to have a portfolio. That means building the foundation for DNDi is building a portfolio so you can choose. The last thing you want is to be stuck with projects, and because they're the only projects you have, you keep going with them. And that has happened even in pharmaceutical industry, where projects have actually been there, and really people should have dropped them, but they stick with them. You have to have choice. So the idea was building the portfolio, a multiple. But one of the questions that came really early with talking to people like Yves Ribet in Synexis and somebody, Anthony Buss, who was in Million in Singapore, when we were discussing how to take on their compounds, they said, so you get a hit. What do you do next? Where do you go? So the other thing that we really had to do as the R&D team was not just build a portfolio, but build a pathway, an end-to-end process, which really is discovery, preclinical, and clinical, to actually take compounds forward. And I think those were the two really essential foundations which my colleagues, the other members of what we call the Three Musketeers, will actually take forward. Tim is D'Artagnan, by the way, just so you get the picture here. So that was really the objective of the discovery. And then it led, in the end, to also the important other part, that each project had to have its own team to drive through, to lead and drive through each project. And that team consisted of chemists, biologists, pharmacologists, regulatory people who know regulatory theories, and that had to be part of the system as well. The teams that could take it from discovery right the way through to development and the clinic. So this was part of the building operation, and I have to say that there were some successes, and I'm very pleased that some of the members who have seen these successes through, which Xing will be talking about, but our first success was actually the work on fexinidazole that Els Terrell led the team on the nitroheterocyclic compounds, led that team, and actually that was the first one that we sort of elevated from discovery into proper preclinical status. So those are really the foundations of how we moved and worked forward. I think in parallel to that, we have to discuss the opportunities that were there. And a lot of these opportunities, again, are around building partnerships that could do clinical studies. There were studies already underway that Eva's already mentioned, but these studies don't just happen by themselves. There had to be the teams built to take forward the nefertimoxythornithine combination for African sleeping sickness, the promomycin combination. I'll mention again the anti-malarial combination in a minute, but also back in 2005 we had early contact with Dr. Sergio Sosa Estani from Argentina, who gave us the introduction to a number of other people in Latin America to start the pediatric formulation studies for Chagas disease. So there was a lot of work being done in this area, building on those opportunities that Yves has already mentioned. But those opportunities, again, cannot just be taken. There has to be, in these areas, properly trained clinical studies, people who can undertake full clinical studies. And one of the key features, which I have to say that Bernard Pecul was very strong on advocating and ensuring we had in place, were the actual clinical trial platforms. And in the period that I was there, the first two, the LEAP platform, which was the East African platform, which was for studies of leishmaniasis, and also the HAT platform in Central and West Africa for sleeping sickness. So those clinical trial platforms essentially meant a lot of training and a lot of engagement at the levels of the clinicians, at the levels of the ministries, and ensuring that the work that was done was focused on the target, as we sort of say, the best science, the best clinical science in this case, for the most neglected. And there'll be more on those later, but just like to say that the other key project that was moving through in that early phase was another formulation study, which it's always easy to say, oh, just stick two drugs together and stick them in a tablet, it works. Actually, the complexity of the project that Jean-René Kichel led was actually showed me particularly a lot of things about physicochemical processes within pharmaceutical research. But again, that was established and moved forward rapidly, and it's good to see some other members of the team here today from Sanofi, and also I think Nick White is here, who's led the sort of scientific reviews of those projects. So actually, when we come to the end of that period, it's just to say a couple of things. This was obviously a fantastic opportunity to establish an international foundation for the rest of the work that the other two musketeers will be talking about in a minute. But it was also not just the international, there was actually the building of the team, and I won't mention I won't mention any of the names of the team, but it was great to actually be back in Geneva building the R&D team. But it's clearly evident to you, I come from a small green island that drifts slowly off northwest Europe. And coming to Geneva was also, for me, a learning experience, not just working with a public-private partnership for the first time, but also moving into a culture and an ambiance which was essentially French. And being an Anglo-Saxon, there was a period of adaptation and learning and developing with the team in this sort of international way. So I think it was the team had to accept me, but I also had to learn how to accept the French culture and the French ambiance. And one of the great achievements of my time there was, I think, having met so many of the team, there was a Jean-René, a Jean-François, and a Jean-Robert, that when I could call myself Jean-Simon by the end of the 3 years, I knew as I resigned I'd been accepted by DNDi. So as Jean-Simon, I hand over to Jean-Xing, who will continue the story.